Traditional molasses and grain distillation can produce up to 15 liters of acidic wastewater for every single liter of alcohol. Today, modern plants operate under a strict zero liquid discharge protocol to eliminate this environmental footprint. We will track this process through 5 stages of transformation. The facility yields 2 distinct commodities: high-purity industrial spirit and a nutrient-dense animal feed called DDGS. In this industrial ecosystem, commercial yield and environmental safety are functionally interdependent. The protocol requires that every liter of waste is processed back into a profitable byproduct. Step 1 is mechanical preparation. Before grain enters the process line, it passes through pre-cleaning. Vibrating screens shake loose dust and straw, while magnetic separators pull out iron and metal debris that may have entered the storage silos. The cleaned grain then heads to the mill. Here, the physical structure of the kernel must be fractured into a highly specific tolerance window. Operators require particles between 300 and 1,000 microns to maximize the surface area for the upcoming chemical reactions. One way to achieve this is with a roller mill. The grain drops through a delivery device into a stacked configuration of textured, cylindrical roll pairs. The opposing rotation of these gears crushes the grain down to the correct micron size. The alternative is high-velocity impact inside a hammer mill. A central rotor spins a series of metal hammers at high speed, pulverizing the grain until the fragments are small enough to pass through the perforated screen at the bottom as a fine flour. If the particle size is too large, the interior starch remains physically inaccessible to the enzymes. If it is too fine, it creates a thick paste that halts the flow of the pumps. Precise milling is the non-negotiable prerequisite for the entire extraction process. Step 2 is liquefaction. The milled flour is pumped into a mixing tank and agitated with hot water and recycled process streams to create a thick slurry. Before adding biological catalysts, operators must stabilize the chemical environment. The slurry's pH is measured and buffered with alkali until it sits between 5.8 and 6.0. At this point, we introduce alpha amylase. As noted in these plant logs under liquefaction, a specific enzyme like Liquiflow is dosed at roughly 0.45 kilograms per ton of starch. This biological catalyst begins severing the complex glycosidic linkages within the starch molecules. This tracks the thermal sequence required to gelatinize the material. The slurry is blasted with pressurized steam in a jet cooker, spiking to 120 degrees Celsius. After a brief hold, it passes through a plate heat exchanger, plummeting to 32 degrees for the next stage. This combination of precise pH, targeted biological enzymes, and extreme thermal shock permanently alters the raw material. The solid starch is now a solubilized, fermentable liquid. Step 3 is fermentation. We start with a 6-hour yeast activation phase. Dry yeast is dropped into the liquid medium and aggressively aerated. The yeast cells hydrate and begin rapidly multiplying to build a massive active culture. This culture is transferred into the main fermenter. We then add a secondary enzyme, amyloglucosidase, to convert remaining dextrins into glucose, along with nitrogen nutrients to sustain the reaction. Over the next 32 to 36 hours, the yeast consumes the sugars. As the organisms metabolize the glucose, they release carbon dioxide gas, producing a liquid wash. Operators track the progress by monitoring the specific gravity. As the heavy sugars are replaced by lighter ethanol, the gravity drops. A successful fermentation results in a final alcohol concentration between 13.5 and 15%. By managing temperature and nutrient levels, the facility uses these organisms as microscopic factories, converting the sugar matrix into unrefined industrial ethanol. Step 4 is distillation. The wash currently holds 15% alcohol, but it is suspended in a bulk volume of water and residual grain solids. Extraction requires separating the pure spirit from this diluted mixture. The wash is pumped through a multi-pressure distillation plant. It moves through a cascade of integrated columns: the analyzer, the rectifier, and the recovery vessels. Because alcohol vaporizes at a lower temperature than water, these columns use heat to boil off and isolate the spirit. To reduce energy costs, the system staggers the operating pressures of each column. High-pressure steam is used in one vessel, and the resulting hot vapor is then recycled to heat the liquid in the next, lower-pressure column. This thermodynamic trick allows a single energy input to perform multiple separations. The result is 95% pure industrial spirit, produced at a fraction of historical energy requirements. Step 5 closes the industrial loop. Once the spirit is extracted, the columns discharge the bulk remainder, a nutrient-heavy, acidic liquid called spent wash. Under the zero discharge mandate, none of this can leave the site as waste. The spent wash is piped into a decanter centrifuge. Spinning at high speeds, the centrifuge strips out the heavy suspended solids to create a thick, wet cake known as Distiller's Wet Grain Solubles, or DWGS. This chart shows the dual path of the remaining material. The liquid is boiled down in an evaporator into a concentrated syrup. That syrup is then merged back with the solid wet cake and fed into a 30-ton capacity rotary tube bundle dryer. The dryer removes the remaining moisture, leaving behind dry distiller's grains with solubles. By transforming the acidic spent wash into a high-protein cattle feed, the facility eliminates its waste stream entirely and achieves the final goal of the grain protocol.